I'm Metin Gürkan. I'm from the Wake Forest School of Medicine. And it's a great honor and privilege for me this morning to introduce Dr. Joel Saltz. He actually needs no introduction. He is one of the most influential leaders in this area. Just for those few of you who have never met him, he is the founding chair and professor of biomedical informatics at Stony Brook. He actually, this is the third time he started the biomedical informatics department. He first started the Department of Biomedical at the Ohio State University, and he hired me over there, and he was the one who introduced me to this wonderful field. And then he went to Emory and he started another department, and then at Stony Brook he started another department. I'm just starting one myself, and I know how difficult it is, and I admire that he can go and do 3 of those things, but he's super accomplished. His group was the first to introduce virtual microscopy in 1997. I just Googled it. It could be earlier, but the reference I found is 1997. Although in Britain, you need to be careful when you say it was first invented. They always say, oh, it was invented 50 years ago by the Scots. There's always another Scottish invention. So there are clinical tracks and technical tracks here. So Joel fits to both of those. He's a pathologist, and then he's also a super accomplished computer scientist, and he can do both of those things. But over the years, he has given a lot to this field. He's actually one of the pioneers and leaders in this area, and then his groups over the years actually have done a lot in this area. And today he is going to take us on a fascinating tour on artificial intelligence, computational pathology, and imaging biomarkers, and we are all looking forward to your talk, Joel.
Well, thank you, Matt. And Nasir, thanks very much for inviting me, and it's great to be here. And the timing of this talk was really quite remarkable. So Jean-Claude JC, who I've known of for many years. We never met, and so this is really exciting and quite an honor to be here. So actually, I don't know if Nasir actually thought. I'm sure you actually probably did to a degree understand how closely these 2 talks would be connected, but they really are incredibly closely connected. So the timing is perfect. So, but we're going to start out with a quiz, okay? Because going from one university to another, you got to have a quizzes, right? Because it's academics. Okay, so AI quiz. So I'm going to give you like 2 ratios, and then people can guess what those ratios are about. So the ratio of a grape, the volume of a grape, to the ratio of the volume of a reasonable sized bear. Okay, so that's one ratio. Another ratio is the diameter of a red blood cell to the height of a giraffe. Any guess as to what that might have to do with AI or pathology? I mean, I'm sure everybody knows. I mean, I'm sorry for being so obvious. Everybody's asleep. Okay, all right. So I'll tell you what the same ratio. Okay, so dating myself, as Metin so kindly did. So I sort of grew up as a MD, PhD, high-end computing person, system software, everything's got to run fast. And actually, I began what I've learned, which is modest, about machine learning and deep learning from Metin. Actually, I hired Metin, and Metin was doing that kind of thing, and Metin and Lee Cooper and a bunch of folks who I hired sort of reversed engineered and figured out sort of how to classify neuroblastoma, and it took years because the methods were sophisticated, but 10 years ago it was much harder then. In any event, okay, so the automagic current methods, like why do they work? I mean, who really knows? But here's a ratio that probably gives some insight. So in '94, I programmed this machine, which was the Intel Paragon, fastest machine in the world at the time, and it clocked in at 143 gigaflops, so 143 billion floating point operations per second. Okay, so the fastest machine now, where we actually have an account and we're submitting a paper to supercomputing tomorrow or Monday, clocked in this year at 143.5 petaflops. Okay, so that is, in fact, a factor of 1 million. Okay, and Jack Dungera, my old chum, who, sort of he does many things, National Academy of Engineering and all of that, but he's very well known for systematically benchmarking the fastest machines. Okay, so he has the top 500 list. So it's gone up by about 1 million. Okay, 1 million. So I was in high-end computing in '94. I was in high-end computing before that, and I guess I'm really not exactly anymore. I do a lot of things, but I hang out with high-end computing. But I mean, it's like a millionfold faster since the '90s. Okay, so I am one of the many people, I will admit, who made fun of neural networks, and I read about them when I was in medical school, like in '81 or '82. I was MD, PhD in computer science. I thought, oh my God, it's the brain, it's so cool, it's so wonderful. And I read Hinton's thing, and I said, oh, this is so exciting. And then, it didn't work. So I get what it didn't work. I mean, okay, so computing and biology, they're totally different. It's silly. Okay, and then life went on, and then, I learned, what I've learned about deep learning from Metin and, all the sophisticated stuff. And then it turns out that, not that you don't need sophisticated stuff, but you can do, amazing things with, really essentially layers of dense or convolutional networks with nonlinear things in between them, and you sort of put them together like, Tinker Toys or Lincoln Logs. This is Inception v4, and I won't bore people with the ins and the outs of this, and I'm probably not the right person to do it anyway. But the point of it is, you go to GitHub, and there are a bunch of these things, and you get them, and they're designed in various clever ways. And in many cases, if you're kind of clever, you don't really have to be that smart. You just have to be kind of resourceful and clever. You can sort of put these things together and do amazing things. So this has huge potential, actually, for curing patients, oddly enough, right? Because basically back in the day, like 10 years ago, you had to get amazing mathematicians to do anything. So it didn't scale. Whereas now you need amazing mathematicians to sort of develop this stuff and to sort of critique and make sure that you're not doing something really stupid. But in terms of day-to-day, you don't necessarily need as many. So you go to GitHub and get these things. Okay, so I'm going to talk about, so I'm going to sort of shift a little bit and talk about what some of the objectives are in digital pathology, at least as I see it, trying to be kind of broad, and then talk about tools of various sorts, both computational tools where the take-home message is the generic is better, and generic often seems to increasingly work. And this is actually, in terms of people who are boundary spanners, this is very good for us, right? In the sense that if you're a computer scientist, and I work with a lot of computer scientists, I used to be a computer scientist, maybe I still am, you sort of have to prove that you've done something really, really clever, okay? And if the take-home message is you doing something really standard works about as well, maybe I'll show some time, describe some methods. Basically we've gone from a really customized method to a much less customized method, like download the thing and train it. It costs 4 times as much time, but is more accurate. And it's okay, so basically you cannot, have to find a new Metin and go for 2 years, and you just download the thing, and it's not optimal, but it takes longer. So live with it. Okay, so as everybody here I'm sure knows, there are a bunch of tasks which have different roles in digital pathology: spatial characterization of tumor and stroma, shapes, textures of nuclei, classifications of cell types, quantitative characterization of lymphocytic infiltration. At some level, these things are building blocks that are just like JC's molecular stuff, right? I mean, and actually what we're doing is reverse engineering or sort of decrypting tissue, right? I mean, so because at some level you can say, here are the cells, what sorts of cells are we dealing with, how are they organized, what's the relationship between tumor and lymphocytic infiltrate, which I'll be talking about. What, if you're talking about GBMs, what are the specific types of cells? So really what you've got is, and I see it 5 to 10 years, and we're seeing this already in all of our groups, the increasing ability to go from pathology as a look at the slide, and this is the name of the pattern, to a multiple level, this is exactly what's here. You still have to figure out what it means and how you're going to use it. So, but it's much more like a molecular thing. It's sort of a little bit like crossing over. Like, I mean, I have these like nightmares of well, not really nightmares, but I mean, I was a very mediocre genetics student. I mean, I was medical school. I mean, I took genetics. I tried to pass. I tried to place out, but I didn't. So I had to take genetics. I did it. It was pass-fail. But I mean, it was okay. But and the problem I always had with genetics, it was all very implicit. And I'm much happier now that you just have the sequence. And RNA-seq, and you've got the sequence, and you have the epigenome, and it's like much more explicit. I mean, figuring out like how many times something crossed over, it's like, I mean, it was so in some ways actually sort of who are the cells and where are they and how are they, it makes it much easier. Okay, so why do we care about this sort of thing? And there are a lot of reasons to care about it, but I'll describe a few. Okay, so I'm in academia, and he and his friends support me. In some general sense, right? Because I don't sell things to the pathology community. I basically live off of grants, I have to admit. And so the major goal of the National Cancer Institute, I mean, I shouldn't say this because I've got somebody right here, but developing new biomarkers to tell who's going to respond to treatment, who's not going to respond to treatment, how to sort of organize therapy. And then you have the related thing involving populations. Okay, so we work with CIR, and they actually fund both a grant and a contract. The grant involving developing methods for taking digital pathology and adding it to information for hopefully all of cancer patients in the world, but CIR handles about a third of cancer patients in the US. And the notion is that there should be digital pathology information, nuanced digital pathology information for essentially all CIR patients. So you look up and CIR has sort of data sets, and if you look it up and you want to say, I'm interested in triple negative breast cancer, and CIR is separately unrelated to us, has a bunch of projects involving tracking outcome and response to treatment, you can say, okay, triple negative breast cancer, let's look at TIL patterns, let's look at infiltrates of lymphocytes, and which patterns do better with checkpoint inhibitor therapy, which no. You could actually, and I think 3 to 10 years, you'll be able to ask that question. I just came back from the CIR-PI meeting, and I mean, there's really great enthusiasm for gathering all sorts of data. And the digital pathology data is the only data that they're going to be able to routinely get that tells you really about biological phenotype. I mean, obviously, there's efforts to get genetic data and genomic data, so I suppose that's unfair. But the thing that's interesting about this is that pretty much all cancer patients have pathology, because otherwise you wouldn't be registered as a cancer patient. So in some sense, there's no additional study that's needed except to scan in the slide and do some computation. So it's incredibly low-hanging fruit. Okay, and so this stuff is really important, and there are a tremendous number of groups, because of the advent of immunotherapy, who are dealing with basically understanding the relationship between tumor and immune interaction, because it's kind of drop dead obvious that if you're modifying the immune system and you already have some status of tumor immune interaction, you're modifying that, and you want to follow how that's going. So that's a sort of very natural sort of thing. The FDA is into this now. The American FDA. I'm trying not to be provincial when it comes to country, and Brandon Gallus, who we're working with, is involved in essentially developing systematic methods for validating what he calls artificial intelligence or decision support methods for FDA clearance, and so he is using. Along with Dr. Salgado and a group of us, and he's very ecumenical, so there are people from all over, essentially going through the process of working out standards for this using tumor-infiltrating lymphocyte algorithms as a sort of prototype for this. So every diagnostic whole slide image, as I've said, contains huge amounts of information. I just wanted to say a few things at multiple levels. And I think a good analysis ultimately will give you information. And it's very similar to what Jean-Claude was talking about in omics. I mean, it's analogous in the sense that you have sort of high-level stuff, which is, what's the classification per slide, per case? You want to ask questions about lymphocytes and tumor regions and how they're related. You want to segment and classify regions. So, for instance, by Gleason score, or actually in the case of Nasir, our joint paper, there's work on non-small cell adenocarcinoma subtype. So things that it's essentially spatial classification using a pathologist context. And then you can actually talk about distribution of glands and ducts, classification, spatial distribution of nuclei, and unfortunately I was at a PI meeting earlier in the week, so I wasn't able to make it to except for today. But it was obvious that people here and people in digital pathology are working on all of these things. And they're not, I don't see these things as being separate tasks. I see it as being one coupled task. I mean, what we want to be able to do, and I think as a field we need to be able to do, is essentially couple these things so that you end up with a multi-scale description of tissue. What are the cells? Where are they? Segmenting them, describing which ones are which, how they're organized in multiple layers, because most questions that you'd want to ask about tissue, at that point, you can ask pretty much what you want. I mean, it's a little bit like if you can do a whole genome sequence versus doing, here are some SNPs, there are some SNPs, there are some other SNPs. I mean, just like we'll be able to do the whole thing. So let's do a whole genome sequence and intensive, expression analysis and whatever. Equivalent here. And in fact, some of the things that I'm real interested in, I know a lot of people are, is the cross product where you're actually doing in situ single cell sequencing, where you're actually looking at expression, mutations, and epigenetics across tissue. And of course, there are NCI projects for that as well, the Human Tumor Atlas and the like. Okay, so I'm going to give a few examples of things that we've done in some of these spaces. So 2 years ago, and one of the tricks that one has to play in this business, and I think that anybody who has an active group in this, sort of does this, if in order to get the computer science, computer vision people to love you, you've got to give them something back, right? And so they've got to get papers like in their good conferences. Okay, so CVPR is one of their good conferences, and one of the things that's fascinating about it, and you sort of I sort of maintain a sense of kind of long-term irony about this, in the sense that you give them something and they have some new twist and it's like, wow, this is really cool. But then I know it's like, really, this is the generic stuff that works. So it's because in some sense, when you think about it, if we're building on what people have done in the last 20, 30 years, and I have a problem, what's the likelihood of the particular person I'm working with today having the exact piece that is going to be absolutely essential for solving the problem? It's much more likely that other things will work, but in order to really sort of keep up to date and be able to use the right thing, you need people at the forefront. So you need to be able to do a bit of an arbitrage. And anyway, so we publish in these places and I think make a nice contribution, but a lot of it is really integrative. So anyway, so one task is, given like TCGA, which is what we actually used, you have a bunch of labels per whole slide image. You don't know why the labels are what they are, and then you've got a zillion patches, so then you want to impute what the classification is. And the paper actually seems to have been referenced a lot. I was happy to see when I was visiting Page AI that their method, Thomas Fuchs is a great guy, and he said, oh yeah, we're kind of using your method for some other stuff. So that was nice. And as an incidentaloma here, as some people here, I'm sure JC notices oligoastrocytoma, which doesn't exist. But it was an interesting thing because that was the label, right? So we went through and did the computation, and you can see the results are terrible, right? Very ambiguous. The confusion matrix, like, it can't tell either. So this was it was banned because of us. No, it had nothing to do with us. It was only coincidence. But in fact, we even knew that this was the old classification. What are you going to do? You get the labels, you run the algorithm. So anyway, so then there was an effort we did with the Pan-Cancer Atlas group, led by Vestian Thorensen, and Alex Lazar is our patron saint of pathology, who is our sort of guiding light on this thing. And so we went through, developed a beautiful and somewhat idiosyncratic TIL classification algorithm, which one of the most important components of which were the pathologists who generated huge amounts of ground truth and the graduate students who developed this thing. So basically, this thing, in some sense, is motivated by some of the manual methods that have been developed by Roberto Salgado and his TILS working group, which I won't go into, but there's sort of a whole set of formulas, recommendations for how you score TILs and tumor-infiltrating lymphocytes, and should it be stromal, which he thinks it should it be tumor, is it close, is it far? Anyway, so Alex was wonderful in working with us and developing a bunch of sort of pan-tumor criteria. These things are they're actually IL maps. These are not TIL maps, they're IL maps because they're infiltrating lymphocytes, whether it's tumor or not, this map. And so here it's surrounding the tumor. Here it's sort of getting into the tumor. We developed a number of methods for characterizing these things in a parametric way, and then joint with the Pan-Cancer Atlas group, we sort of related these things to molecular data, to outcome, to the distance to the nearest extrasolar, planet or whatever. I mean, the TCGA analyses tend to have this encyclopedic thing, which I think is really good because now there's a bunch of data that can be analyzed by people, including us. So I've got some MD, PhD students sort of taking deeper dives into this, and some kind of intriguing things involving relationship between molecular subtypes that the immunology molecular group came up with in patterns of lymphocytes. Okay, and all of this is publicly available. And then this got rolled into the sort of more broader publication, and it was all in like some wild and crazy splash that Jean-Claude sponsored in Cell. So zillions of papers going out. So this got rolled into sort of relationship between molecular and spatial patterns. So sort of a point getting back to it. So the algorithm, I won't bore you with, but it was a very clever algorithm, and we got some nice publications out, and now it's outdated. Okay, I mean, I think one of the things in this field that's really fun, because I tend to be easily bored, frankly, is this holding on to, it's my algorithm, I love my algorithm, you better not touch my algorithm, it's a secret algorithm, it's ridiculous. I mean, so basically, a year later, turns out Inception V4 and VGG with really good training data, because the process of this, we generated a lot of training data and it works better, actually. So the magic algorithm is no longer really that important, basically, and so we're making our trained models public, along with the code, which is already public because it's based on standard stuff. So we went through like, a mini Metin experience, maybe call it 1/3 of a Metin, doing this thing. And now it's like totally generic. So anyway, so let me wrap up here and make another related point, nuclear segmentation. Okay, so this is a sad picture. Okay, so why is this a sad picture? Well, it's not sad, but it's boring. Okay, so 4 years ago, we had a level set mean shift algorithm that another wonderful mathematician, Alan Tannenbaum, and his sort of student who was for a while on my faculty, Yi Gao, developed. And it was wonderful. And as far as we could see, it was as good as some of the other ones. I mean, we benchmarked it. And it was terrible, really. I mean, basically, you had to adjust the threshold. And basically, what this thing is software for putting together a mosaic of regions where different parameters give you good results. And we were doing this for SEER. So we had all these grad students sort of curating this stuff. And it was like, several hours per whole slide image to get like a decent result. I mean, you could run the thing, but in order to get a decent result. So now we have, a wonderful GAN thing, another CVPR publication. And in fact, Nasir's group, we did a McKay, actually, Tassenkirk, one of my faculty collaborators, did a McKay competition. And of course, there was a wonderful deep learning nuclear segmentation as well as region classification method coming, from here. And really, basically, the stuff more or less works. I mean, frankly, the brand of it, I'm not convinced is that important. It doesn't work perfectly, but I'd say it works much. I mean, it works well enough that we ran our thing on 5,000, the same 5,000 slides as in the Cell Reports paper, and we're making it public. I mean, so it's not perfect, but it's good enough. It's more analogous to gene sequencing. It's it's not like 1,000-fold coverage or something, but I mean, it's not bad. Okay, so we have a bunch of tools, and I want to say in particular, since I have JC right here, there are lots of tools for managing nuclear and spatial features of pathology data, and the GDC is wonderful, but that's not where it's supposed to live. Okay, and I know you know that because, of course, there's a whole imaging data commons thing and all of that. So I mean, there's a lot of things that, fortunately for you, I don't have time to get into, but there are lots of tools. We're involved, and I'm a multiple PI of an extension to the TCIA to oversimplify, which involves pathology and pathology features. We have actually a very nice metadata management system that we just rolled out like a week ago, where you can look at things like TIL maps and zoom in and look at particular regions. So it's a heat map of cancer, heat map of TILs. It's generalized, so you can look at nuclear features and you can zoom in and look at particular areas. But the thing that's cool is you can sort of, and it's actually online, so I can show people. I just didn't have time. So you can click in areas that have a lot of one thing or another, and then look in the whole slide image and see where it is. So there are lots of projects that use these sorts of things. So anyway. So in conclusion, and this is really, I mean, for boundary spanners, which I think most people in digital, I mean, if you're in digital pathology, you kind of have to be, right? What else are you? This is really an incredibly fun period because the world is producing toys for us. We can use them. It's people with sort of broad skill sets, like us, are really in a tremendous position to put together some very powerful projects that in the past would require a huge budget and an ability to herd cats that is difficult to sustain. So I think it's a lot of fun, and this is really, I think obviously an extremely. I'm excited and honored to be part of Nasir's grand coalition. This is fun, and I think there's a tremendous potential for interesting things, and actually for joint work across the Atlantic and the Pacific, and to really make an impact on cancer and really help some people. So lots of people involved. This is the SEER team. This is our ITCR team. Funding from various NCI sources. So I want to thank everybody, and happy to take questions to the extent time allows.
Let's thank Dr. Saltz. Any questions? So I don't know if you had a chance to pack your crystal ball, but if I can ask that crystal ball question. So just some of the stuff is just obviously 5-10 years it will take us, but where do you see things in 20-30 years?
Well, it's very hard to know. I mean, if you assume that there isn't progress in actual medicine, which at that time scale hopefully there will be, then obviously if you took sort of the current medical practice and the current medical knowledge about cancer, and you said, okay, let's say you had all of the things could be done technologically in, say, 20 years. And so let's say you're a pathologist, and you say, okay, here's tissue, and what you'd be able to get would be essentially data products that would help you make decisions based on knowing what and where all of the cells are, right? So it's not really so you're in a position, if there is research that says, well, a certain pattern of lymphocytes or a certain pattern of tumor cells or a certain pattern of inflammatory reaction or whatever is important, then you could get the data sliced and diced. So it would be a little bit like working for Google, which I've never done, in the sense that you have data analytics that would support the questions you want to ask. Then from the research point of view, when you develop biomarkers, you'd be able to ask, as you do now in omics to a large degree, what patterns are important. So the pathologist, sure, they could look at the image. How often they do it, I don't know, but I think it would be important to. It's sort of like a pilot, But then you'd also get a lot of summaries. So with current state of medicine, I think that actually pathologists will be doing more rather than less, in the sense that you'll have much deeper data to sort through. Now, obviously, realistically, in 20 years, hopefully we'll have actually made some progress towards treating these diseases. So a hope for sort of thing is if mostly you're able to manage cancer as a chronic disease, or even sometimes get rid of it till you die of something else, then it would be really of a question of, okay, it's kind of a cockpit. It's like, okay, this is beginning to happen. What sort of treatments make sense? So it might not be as integrated. It might not be as siloed where now you've got a pathologist and you've got an oncologist as separate people. You basically have one person or a team saying, okay, what are we doing? Here's the radiology, here's the pathology, here's the clinical, here's the omic. I mean, because that's what we're all trying to do, is put it together. So in some sense, having these expensive silos giving you little pieces of things and then having some other person say, here's the report of this and here's the report of that, in some sense, the ability to integrate this stuff, which is also of tremendous interest, I think if you give 20 years, it'll actually, to a degree, work. So it would be a little bit like flying a plane. And when you think about it, I mean, flying planes usually works, and there'll probably be, occasional disasters. It's probably not 100%, but so that's my guess.
Excellent. Any quick questions? Yes.
Just a comment regarding your parallel between 94 and now, the million difference between the computing power. Also, the data changed, not so much, but a lot, because in 94 we didn't have digital slides. Some of the things you mentioned, and everyone is mentioning, like nuclei features, spatial distribution of nuclei, etc., were made in 94, but on single images, which are, let's say, 100,000 less than the data we have now. So we were doing what was called morphometry, or at that time syntactic structure analysis, but on single images, losing the view on the full slide, which gives extra information not available at that time. So the parallel can be done also on the data. One image versus the equivalent of 100,000 images for a single case. So it's something to consider. We have more power, but also more data to deal with.
No, I couldn't agree more. And actually, so in 96 or 97, we got an amazing machine from IBM. They gave it to us, an SP2, and it had 202 gigabyte disks. And they claimed it was $1.5 million. And in fact, we bought a Bacchus scanner, which took photomicrographs, and we programmed the thing to take all photomicrographs. And the deal with IBM is we were also doing land cover stuff, that this was sort of an experimental system. And I gave grand rounds at Hopkins in 98 and said, I was a little nervous because I was just kind of new there. And I said, it's going to be 20 years before this thing works, because who can afford the data? It's like $10,000 a whole slide image if you do such a thing. Then I later found out the Bacchus people were actually quietly doing their own patent. We were academic type. So it's like, I just found that out actually a couple of years ago. It's like, gee, they were doing this too, but they didn't tell us. It makes sense that they did, because actually you could program their machine to do it, so they weren't such idiots that they didn't realize that. But no, I totally agree. And I also agree, it's very much like neural networks. Most of the stuff isn't new. I mean, a lot of it is just driven by the improvements in storage and computing capabilities, so you can actually scale them. So I completely agree, and I totally am with the, we're inventing X. Nobody's done X before, and it's like, okay, you guys were doing X like in 1971. So yeah, I'm with you on that.
Let's thank Dr. Saltz one more time for his great talk.
Thanks.